Hi, my name is Kyndal Irwin. I am with Texas State University. I'm so excited you tuned into this seminar. Hopefully it breathes some life into my most recent publication in Biology Letters regarding a very sophisticated interaction I recently discovered in which habitat complexity predicts cognitive performance and behavior in Western mosquitofish. So cognitive ecology is a field of science meant to untangle what aspects of the environment are shaping animal cognition and behavior. And my research interests in the field were largely inspired by my environment at Texas State University, where I am a PhD student. So our campus is very unique and located in Central Texas, and it's beloved by Texans because the San Marcos River runs immediately through campus. So here's a photograph of Sewell Park. This is just one portion of the river that Texans and students love to frequent to cool off and find some relief from the Texas heat. It's truly a beloved source of recreation, education, and conservation efforts, and it's at the heart of San Marcos culture. So the San Marcos River is an abundant source of biodiversity. It's home to some critically endangered species such as the Texas blind salamander, definitely a crowd favorite, and also Texas wild rice, which is an aquatic species of plant and almost moves like mermaid hair through the water. And so while I'm presented with this just precious and abundant source of wildlife running right through our city, I can't help but feel an enormous sense of gratitude and also a sense of curiosity. So as much as we love the river and our proximity to just this absolute treasure trove of wildlife, how is the wildlife responding to us? So a major question that I want to know is, how are animals responding to human alterations to their environment? So previous literature has shown that urbanization and subsequent increases in impervious cover lead to the physical and ecological degradation of streams draining urban land. So impervious surfaces like cement prevent the reabsorption of runoff back into the ground, and instead, there's a huge volume of runoff running into urban streams. And this causes altered hydrological regimes, increases in contamination loads, because with that runoff, you're getting a lot of litter, other contaminants like plastics, and also reduced physical habitat. So with those increases of volume of runoff, a lot of urban streams are scoured. There's altered channel morphology and sediment deposition that actually limit which vegetative species can grow there. And so overall, we see lower levels of biodiversity in urban streams and ultimately lower levels of habitat complexity. And so as a researcher, I'm really interested in how animals are responding to such anthropogenic changes, these human alterations such as urban stream syndrome, which is the term used to collectively describe the symptoms that urban streams are suffering from. How are animals responding to these, right? How are animals responding to urban stream syndrome, and how is this affecting their cognition and behavior? So what is animal cognition? Cognition mediates the perception of environmental stimuli. Cognitive abilities show variation across populations and individuals. So this indicates that natural habitat directly influences an organism's ability to learn, make decisions, and retain memory. So these are all components of cognition. And cognition not only mediates perception of environmental stimuli, but it also modulates behavioral responses that are important to an organism's overall fitness. So cognition is super closely tied with behaviors like risk-taking or the willingness to explore novel habitat. And behaviors like this can be really important to survival, invasion success, occupying new habitats, and so on. So cognition and behavior are very closely tied. In various situations, animals have to modify or block automatic responses and switch to diverse, more appropriate responses to achieve a certain goal in a certain context. So in biological terms, the cognitive function responsible for this is referred to as inhibitory control. And a component of inhibitory control is motor self-regulation. And it's used to describe the physical response to the alternative decision-making. So I'm using this language, but essentially we exercise inhibitory control all the time. It's regarded to as self-control, right? So some people might be more familiar with that concept, and it's essentially the same. So there's an automatic impulse or response, but this initial reaction isn't ideal to accomplish a given task. So we pause and actively engage in an alternative behavior that will allow for a more efficient outcome. So we know that cognition and behavior are closely tied. How is this related to the environment? Well, cognitive processes may be more or less flexible according to the environmental demands in which a given animal is living. So we know that different environmental demands exist in different habitat types, and this can affect the selection of cognitive abilities because we do see variation in cognitive performance across populations and individuals. And this is exhibited in differences in brain size, invasion success, foraging behavior, and overall fitness. And so this intimate relationship between the environment and cognitive ability gives rise to the cognitive buffer hypothesis. And the cognitive buffer hypothesis is a notion that in more complex environments, there is a greater abundance of environmental stimuli to engage and process, and therefore animals in more complex environments are likely to select for greater cognitive abilities to deal with greater information loads in those habitats. So I decided to explore the connection between habitat complexity and cognitive ability and behavior in Western mosquitofish, or Gambusia affinis. So the Western mosquitofish is a widespread species of freshwater minnow. They're native to Texas, but they have spread to over 40 countries where they are considered invasive. They were introduced as a means of biological control to deal with mosquito populations because they're very voracious eaters, but turns out they eat a lot more than mosquitoes. They're incredible predators, and they often outcompete sensitive species. So they are a tolerant species of fish. They actually thrive in urban areas. They're shoaling species, so they're very social and rely on their shoals to reduce predation pressure. They're also a live-bearing species of fish, which is very interesting. So they don't lay eggs, they give live birth, and at any given moment, a female can have 100 babies at a time. And these fish also occupy a wide range of habitat types. So they're mostly of interest to me and other conservation biologists because of their tolerance, right? So by studying their cognition and behavior in the context of their environment, we can gain insight into what makes this particular species so successful in navigating human-altered landscapes, while others are not. So as I said before, mosquitofish live almost anywhere, and different populations exist in various habitat types, ranging in environmental complexity. So environmental complexity is a term used to describe the level of heterogeneity on spatial, temporal, and biological scales. But in our study, we defined environmental complexity as the variation in physical habitat structure with greater heterogeneity equating to higher complexity. So mosquitofish can live in low complexity environments with flat, muddy substrate and very few plant species, to very complex habitats with elaborate limestone structures and a variety of different vegetative species. So earlier I was talking about urban stream syndrome, and a common symptom of urban streams is that urban streams tend to be on the lower end of the spectrum for complexity, but mosquitofish can persist in super structurally complex habitats as well, and navigating through these spatially complex habitats, there is additional information loads, right? Keeping track of resources and evaluating predation risk is cognitively demanding and requires processing and storing large amounts of useful information and filtering out background data. So building on the cognitive buffer hypothesis that I mentioned earlier, I hypothesized that environmental complexity affects the cognitive performance and behavior in mosquitofish, and I specifically predicted that fish from highly complex habitats would exhibit greater cognitive performance and riskier behavior compared to fish from low complexity habitats. I predicted that they would show relatively lower cognitive performance. So to test my hypothesis that environmental complexity affects cognitive performance and behavior in the species, I went to 9 different locations throughout central Texas where I identified Gambusia finis, and I measured habitat structural complexity using an optical analysis technique adapted from Shumway et al. 2005. I also measured the degree of urbanization for each stream by quantifying the degree of total percent development of the stream subwatershed in ArcGIS. So I took the total percent development and I calculated that based on the amount of impervious cover. And then I collected 35 to 40 adult female Gambusia finis from each site to be compared in a laboratory experiment where I measured their inhibitory control as a measure of cognitive performance, inhibitory control specifically motor self-regulation. And I also measured their risk-taking as a behavioral measure, and I compared these scores across 9 populations in a detour test in the lab. So here's an example of how I captured habitat complexity. So I laid out a 10 by, like a 10 meter transect where I found the fish. And at 6 equidistant points, I recorded videos facing upstream using the 0.5 angle on my iPhone and a waterproof sleeve. So I used a color wheel in the first part of the shot, and this helped me standardize later on when I did adjustments in iMovie and ImageJ. But here's an example of what one of our videos would look like. I took 6 at each habitat. So I'm capturing sediment, vegetation. Actually, you can see Gambusia shoaling off in the distance, right? And so from each habitat, I took 6 of these videos, and then I uploaded them to iMovie, and I randomly extracted 18 frames from these videos. So then I converted these frames to 8 black and white bit grayscale and actually quantified habitat complexity based on the variation in pixel intensity. So each pixel in that photograph receives a numerical intensity score. It could be 0 being totally black to 255 being totally white. And by our standard, by our definition of environmental complexity, right, higher complexity, greater variation. So I took the standard deviation of these pixel intensities. The greater standard deviation, the higher variation, the greater the complexity, right? So that's how we actually quantified the complexity of each of our sites and gave it a numerical quantification. And so then I placed each of my 9 sites in one of 3 categories, either low complexity ranging in a normalized value from -1.17 to -0.37. A site could be moderately complex with a range from -0.29 to 0.31, or highly complex, right? 0.83 to 1.67. So these are all actual photos of some of my sites and the photos that I used for this analysis. And so now that I've quantified habitat complexity, I wanted to compare the fish's cognitive ability and behavior in a detour test. So this is a graphic of what the detour test looks like. So on the left, we have a focal female, and she is in a covered starting chamber where I acclimate her for 10 minutes. And then on the other side of the maze, we have a reward fish. So as I mentioned, Gambusia, they are shoaling species. They're very, very social. And so a conspecific individual is an appropriate reward in this test. So I put my reward fish in a glass jar on the other sides of a maze. So after this 10-minute acclimation period at point A, I lift open a gladiator door. And the time it takes the female, the focal female, to exit the starting chamber into the testing arena is how I measured boldness, risk-taking behavior, or exit time. And so that's label A, right? Arrow B is motivation. So the time it took the fish to exit the chamber and then reach the gray plexiglass barrier. Now, once the fish reaches the plexiglass barrier, she can see the conspecific reward, all right? But this is the point at which solution speed or motor self-regulation is actually elicited, because the direct pathway to the reward is blocked. So in order to reach the reward, the fish has to travel through pathways marked C. There are actually gaps in between the wall and the plexiglass that the fish can navigate through. But at that point, there's a boundary, right? And she can no longer see the reward. So there has to be an element of object permanence to be able to successfully solve this maze and reach the reward. So as I was testing these fish, I actually noticed some significant differences. But let me back up a little bit. So exit time is how we measured risk taking. Motivation, right, is B, and then solution speed is how we quantified cognitive ability. And while I was testing these fish, I already noticed some really striking differences. So here's a video of a fish from a highly complex habitat. So I've listed the chamber, she exits. She sees the conspecific female. She's kind of hesitant. She reaches the plexiglass barrier, right? So she might swim back and forth across that plexiglass barrier for 4 minutes. The fish have a total of 10 minutes to actually solve the maze. We gave them a max score. But here is a fish from a low complexity habitat. Play it. So I lift up the door. Boom, super fast. This was actually my fastest fish. She's very smart, A+. So we ran a Cox proportional hazard model with sensor data based on whether fish exited or not. That way we could account for solution speeds. And we did this by complexity level, low, medium, and high as the factor, with standard length as a covariate and population as a random effect, with a significance level set at 0.05. So here on the y-axis, we have exit time, and on the x-axis, we have complexity level. Each point represents an individual fish, and the letters indicate significant differences between groups. So we found that fish from low and medium complexities were faster to exit the starting chamber compared to fish from the high complexities, which indicates that fish from the lower complexities are exhibiting riskier behavior. So here we've got motivation and motor self-regulation. On the left, we've got motivation on the y-axis, and again, complexity level on the x. And what we found was that fish from low complexity habitats were significantly faster. They had faster motivation scores, so greater motivation compared to high complexity habitats, with medium complexity not being significantly different from either low or high. But we found the same pattern for motor self-regulation. So again, opposite to our predictions, fish from the low complexity habitats had faster motor self-regulation. They were faster to solve the maze compared to fish from the high complexity sites. So it's pretty exciting when you find something totally opposite from your predictions. So what do we take away from this? Is that fish from urban sites are smarter? Well, not exactly. We did not find a relationship between habitat complexity and percent development, but we think that this is mediated by stream order for our sites. So we did find a relationship between habitat complexity and stream order. So stream order is the numeric classification of the relative stream size and river branching, with the smallest tributaries referred to as first order. And so we ran a Spearman's correlation and found a negative correlation between habitat complexity and stream order. So here, right, normalized habitat complexity on the y, stream order on our x. And habitat complexity was greater in first order streams in our sites and low impervious cover watersheds for one of our sites. but also high in impervious cover watershed. And so first order streams with high habitat complexity independent of urbanization are actually really common among Central Texas streams that originate from perennially flowing springs fed by karst aquifers. So water permanency is greater near first order streams and then decreases with higher order streams. And we see occasional drying during drought or seasonally drying during summer. So as a consequence, first order streams tend to have more habitat complexity than downstream and higher order stream reaches. Furthermore, right, watershed size of the first order streams with perennial spring flows tend to be small due to the mechanism of spring outflow formations. And therefore, they're protected from flashy flows caused by periodic rain events. And in contrast, downstream and higher order streams are more susceptible to flashy flows, which reduce habitat complexity caused by periodic rain events and this is exacerbated with increases in impervious cover. So back to my favorite part, the cognition and behavior, right? Habitat complexity affects cognition and behavior. That's our big take home. We found that mosquitofish from high complexity habitats were less risky. They showed lower motivation scores and lower motor self-regulation. Whereas fish from the low complexity habitats were more risky. They showed higher motivation and greater inhibitory control. So what exactly is going on here? I think that this pattern could be driven by predation pressure. In low complexity habitats, there's less refuge available for fish to hide from predators. So it's likely that in low complexity habitats, there's higher predation pressure, right? So this could then select for fish that have to take greater risks, have higher motivation and greater motor self-regulation to be able to navigate through their environment and stay with their shoal. So of course this would have to be tested in a future study, and whether the effects of habitat complexity on cognition and behavior are plastic or genetic are not known yet, but I'm actually starting a follow-up experiment to ask just that. And so with that, I would love to acknowledge the GASP Lab for all of their help, all the GASP Lab members, and Texas Ecological Laboratory for funding my research. And of course, I want to thank my advisor, Dr. Gabor, who's been the best science coach ever. I love being her student so much. I got my master's with her and I've stayed for my PhD. She's just my science mom, and she's really, really helping me explore my interests. And I just really appreciate her support. So thank you so much for watching. If you feel like contacting me for questions or if you want to do some science together, you can contact me at kbi4@txstate.edu. Thank you so much.